cloud. All right, well, welcome to the latest installment of the Coronavirus Multi-Species Reading Group. It's a great pleasure to have Konstantin with us today. You've offered us a paper that's just about a year old, and I think it offers a really interesting perspective. You know, this is a paper titled "Can Live Vaccines Prevent COVID-19?" written again at a very different moment where we didn't have standard vaccines, and I think it speaks to broad concerns in our field, multi-species studies. So there's a lot of work over the years about probiotic bacteria, and I think there's many people out there, including kind of commercial interests, that sort of celebrate microbial life and sort of look at the ways that we might develop a mode of being in the world that's not just Pasteurian, sterilize everything, but we might selectively engage with microbial communities for generative possibilities. So a number of anthropologists and others have written about what Heather Paxson calls microbiopolitics. That's basically ways of engaging with microbes that don't just sort of welcome all, bacteria or viruses, but sort of, again, selectively think about in a particular situation, how another form of life might be generative, not just pathogenic. So I think this piece does really interesting work to get us thinking. You're broadly talking about oral poliovirus and the unexpected health benefits that were conferred to people who took this. You're going through the evidence, looking at everything from influenza viral infections to genital herpes, as well as oncolytic properties, the ability of these viral infections to change how the body responds to cancer. And maybe I would love to just start with an update to hear from you. Now that we're a year on, do we have any sense of so here you're predicting that if someone has a live attenuated virus that isn't necessarily a coronavirus, but maybe something closely related, that's going to increase the likelihood that they're not going to get seriously sick with COVID-19. So now that we've got a vaccine for SARS-CoV-2, now that we know more about how SARS-CoV-2 works and how the disease of COVID-19 unfolds, what's your latest thinking about live attenuated viruses? And do we have more evidence? Looking back from where you were sitting a year ago.
Well, look, I mean, you've mentioned our paper that we published with Bob Gallo and a few other colleagues in Science. I think it was June of last year. So yeah, exactly a year ago. But since then, we also published just recently, I mean, maybe a few weeks ago, another paper, more expanded version of this, because that one was basically an opinion paper, 2 pages. This one is more expanded. I've just sent it through the chat. So you can download it. This is a paper where we basically provide a little more scientific justification, because the first one was more or less kind of announcement of that this idea could be explored. At the time, we were thinking about setting up a clinical trial of this idea with OPV. In fact, Bob Gallo of the Institute of Human Virology, and I think he does not need introductions. He's the discoverer of HIV virus. So we were working together, trying to set up this trial, applied to NIH for funding. But of course, at the time, everybody was so, single-mindedly focused on creation of specific vaccines. And this Operation Warp Speed was created in the United States. Billions were spent on these vaccines, were turned out to be quite successful. In fact, I think at the time we did not expect, and in fact, in this paper, we were sort of wondering whether it is possible at all, how long the immunity will last, and so on. and what would happen if virus starts drifting, just like flu virus, for instance. But I mean, we failed because of a number of reasons, mostly not scientific, but rather, outside. I would maybe even could call it political. But one way or another, vaccines were introduced at the end of last year, emergency use authorization, Pfizer, Moderna, Janssen, and so on. And they're now used quite widely, and they are really successful beyond anybody's expectations. But now, I mean, as you know, the reports trickling in about this new strains, this Indian strain, Delta, whatever they call it, that based on the lab data, it sort of partially evades immunity created by the old vaccines made against the old Wuhan strain of the virus. So, and this is something that we were talking about, that it could happen that at some point we will maybe need to change the vaccine to something, update, do it based on another antigens. And with some vaccines, it may not be that easy because, for instance, if the vaccine is made using adenovirus vector or another vector, second, third, and subsequent doses will be decreasingly effective, I would say. So because immune response to the vector itself will block vaccine potentially. So that's why having a broadly specific, a broadly protective vaccine would be really, really important. And maybe not even for this pandemic, because right now it looks like we do have some effective tools against it. But suppose a new pandemic comes. So we will have to start from scratch. First, we'll have to identify virus. Second, develop the vaccine. And I mean, 1 year is probably the best we can do. And during the first year of a pandemic, countless lives will be lost. And this virus turned out to be not the worst of all. I mean, we were lucky because, I mean, of course, several million people passed away, but still, it could have been much, much worse. And the vaccine could not be as successful as it is. So right now, I think in our pandemic preparedness, there's a huge gap. We don't really have anything to cover for the time period where new vaccines are being developed and produced, tested, and rolled out. So I think that this is still, I believe, is very, very important aspect. And the history of this is that I was always fascinated by this phenomenon that was discovered many years ago, that vaccines protect not only against target pathogen, because of course, I mean, they are designed to induce humoral and cellular immunity that specifically addresses the infection that it is created against. But perhaps by inducing innate immune response, broadly protective pathways in our body, that include a number of things such as interferons that are called such because they are induced by viruses to interfere with another virus. Basically, they induce in our body, they induce the so-called antiviral state, so that the organism becomes less susceptible to other infections. And there are multiple different biochemical pathways by which it is accomplished. And it also includes a number of cellular components, both lymphoid and myeloid cells, such as macrophages and natural killer cells, and so on, so that they are also activated. And they act in concert to suppress viral infection. So I was always excited, and unfortunately, very little studies are going on. I mean, there are increasingly more, but I think that most people, when they talk about vaccines, they mean only antibodies, and some of them also know about, T cells, and that's it. And they look at innate immune response as a kind of auxiliary mechanism to, because induction of innate immunity helps adaptive immunity. I mean, it's a part of the body's response and the generation of antibodies and T cells and so on. But in fact, innate immunity, if you think about it, it is probably by far more important than adaptive immune response. Because adaptive immune response develops as a minimum, it requires a couple of weeks minimum, maybe 10 days, but more often 3 weeks or even more. And during this time, we have to survive somehow. And if you think about your the viral diseases that you had in the past, be it a flu or rhinovirus, common cold, usually we come out of it in 3-4 days, right? You have fever, you have chills, muscle aches, you drink hot tea, and that's it. You recover even before your immune response have a chance to develop antibodies. So we don't die. And just because our innate immune system starts working immediately. The minute and hour we are infected, all these immune responses that are broadly specific come into work. And as a result, our body clears this infection pretty quickly. And then, of course, as a consequence, after the fact, it develops memory. It develops antibody that would recognize if next time this virus comes back. So then it will deal with it even more effectively. So, but essentially, the reason why we're still alive is because of the innate immune system. And this was, of course, confirmed when this SARS-CoV-2 was discovered and studied. It turned out that from many lines of evidence, it became clear that this virus, just like many other viruses, it is mortally afraid of innate immune system. First, it carries several genes that specifically target innate immunity pathways. So it wants to turn it off. And if you inactivate these genes, the virus becomes attenuated. So in fact, for pathogenicity of the virus, it is critically important for it to suppress interferon response pathways and several other pathways. So that's one evidence. The second evidence comes from the human genetics, genomics, because when they have done this analysis of severe cases of COVID and performed this association study, you know, by sequencing or genotyping human genome, it turned out that the only thing that correlates with the severity of coronavirus disease are genes responsible for innate immunity. So people who have these pathways damaged somehow genetically, you know, impaired, they develop severe disease and die. So these two facts together, they suggest that this is critical. And since the virus has this way of switching off our innate immune response, then it's logical to boost somehow. I mean, to provide another virus before or maybe even after infection that would compensate for this defect. That's actually how we came up with this idea that using other live attenuated vaccines could help in this particular case. And of course, like I already mentioned, I think that it has a much broader implications for pandemic preparedness than just dealing with this pandemic.
Maybe before you go into the idea of the live attenuated virus, you could just slow down a little bit more with the innate immunity. So you mentioned the interferon pathway as one important pathway that SARS-CoV-2 interrupts. Could you just sort of spell out the basics for someone who doesn't know anything about interferons, and then also talk a little bit about the other pathways that SARS-CoV-2 interrupts?
Yeah, I mean, interferon, it was discovered in the 1950s, when this phenomenon of viral interference was discovered, and it was both in vivo and in vitro, it was found that if you infect with one virus, then you cannot superinfect with another. So there was a way of viruses to basically fend off each other. And the studies revealed that it is done based on the induction of a protein that's called, that's the reason it was called interferon. But, after many similar proteins were discovered later, I mean, it's a cytokine. I mean, it's one of those proteins that our body uses for communication between cells. Cells, they secrete these cytokines, and they go into the circulation, and they bind specific receptors on the surface of other cells and send a message. So in case of interferons, and there are several kinds of interferon, alpha, beta, gamma, or type 1, 2, and 3. I mean, and some types are even subdivided into many other. It's a family of genes and family of proteins. So what they do, actually, they induce the so-called antiviral state. And they do it by binding the receptor that triggers a cascade of events that leads to transcription of several proteins that are called antiviral proteins. And they do different things. Some of them, for instance, phosphorylate proteins that are involved in protein synthesis. And by doing this, they shut it down. Cell can't survive without protein synthesis for a while. I mean, it has enough, but virus will not survive. It will die. And as soon as the virus is dead, then this inhibition actually goes away, and the cells resumes its normal life. Another of these antiviral proteins, it induces a nuclease. First, there is a special enzyme that produces the short oligonucleotide 25A. It's a trinucleotide made from adenosine residues, but it's connected not through 3,5 phosphodiester bond, but 2,5. It's kind of weird oligonucleotide, and its role is to trigger a nuclease that would digest all ribonucleic acids. So basically, it's a mechanism for a cell to burn the house down. I mean, you just wipe out all nucleic acids. And again, for a cell, it's not a fatal thing. It can recover, because, I mean, the moment virus is purged from the system, then it starts making new RNA from DNA and so on, and it will come back to life. So there are many other mechanisms. I cannot talk about them all. I don't even know them all. But one way or another, cells and the organism as such, it becomes resistant to viral infection. Not only by the same virus, but by many other viruses. So this is a kind of more or less well-known aspect. And another cellular aspect is much less studied. And again, it involves a lot of different cells, but the idea is the same. As soon as the body detects foreign invasion, and it's done by the special receptors, so like a TLR receptor and NOD receptors, there are and they're called pattern recognition receptors. Basically, they recognize chemical groups, chemical patterns that are not supposed to be present in a normal cell. For instance, many viruses replicate through a double-stranded RNA intermediate. And normally, cells don't have double-stranded RNA. And there are receptors that recognize double-stranded RNA, and they trigger this cascade of events. There are pattern recognition receptors that recognize special bacterial polysaccharides and also respond. So there are multiple receptors. That's basically sentinel molecules that look around and try to find something that is foreign. And that's how they work. And the cellular activation of cellular mechanism is going through the same way by activating transcription of genes that induce this increased viral resistance and ability to clear out viral infections. And the interesting new emerging field is the so-called trained innate immunity. It's not just like activation of some specific, broadly specific mechanism, but rather it's kind of sort of immune memory, because if a cell was assaulted by a virus, it activates genes, and gene activation often includes methylation and acetylation of histones, proteins that basically make the chromosome. And by doing this, it basically loosens up the chromatin and makes it easier for transcription to take place. It turns out that if you expose cells once to this injury, then it remembers, because this acetylation and methylation stays. And next time you apply similar stimulus, it will do it much faster. So basically, it's kind of priming the immune system for a rapid response. So that's why this non-specific protection sometimes can last months or maybe even longer. So it's not like a few weeks during which interferon is circulating. So potentially it could be really an interesting and very useful way of dealing with viruses.
Super interesting indeed. And I might ask, Amina's got a couple of questions in there about cytokine storms as well as reception of the scientific community. Amina, do you want to go ahead and ask them yourself and unmute?
Sure. Yeah, I have it in the chat. Yeah.
Yeah.
So, yeah. Please go ahead.
So 3 questions here. Pick as you like. One, I'm just wondering about how what you're saying relates to the early ideas that we were hearing and concerns about the coronavirus overstimulating an immune response. So rather than suppressing it, it was causing these cytokine storms, especially for people who had seemingly overreactive immune systems. And then along a different line of thought, Evan mentioned something about probiotics, and I'm wondering if there's a way of framing the live attenuated vaccine as something like a broad spectrum probiotic. Is that inappropriate because it's attenuated? And then I'm just wondering about the reception of your proposals. Do you see any pushback that is, from your perspective, coming from a non-scientific, not a scientific instinct, but some other sort of influence?
No, I mean, yeah, let me first answer the first part about the cytokine storm. And yeah, indeed, of course, hyperinflammation is it's a kind of important part of COVID-19 pathogenesis. And I'm not sure what will happen if you stimulate the system even further. It remains to be seen. But what our idea was that we use it prophylactically, or either giving this live attenuated vaccines to normal healthy people who are not infected with COVID, or somebody who's been just infected and still is incubating, kind of a post-exposure vaccination to prevent them from progressing from asymptomatic phase into symptomatic phase. But in fact, there are some ideas that activation of viral activation of these pathways, actually it suppresses inflammation. There are some studies conducted not with viruses, but rather with attenuated live bacterial vaccines that demonstrate that this inflammatory diseases, including non-viral, non-infectious inflammatory diseases, are suppressed by the use of this live attenuated bacteria. So, I mean, this is a good question, and of course, we need to be careful. But I can see the situation where this could be even helpful, not only exacerbating cytokine storm, but maybe even helping to reduce it. Now, your question about the probiotics, it's a very good one. I like it. Because, I mean, we all know probiotics, and we all know that those bacteria that populate our gut, they have a tremendous role in our immune system. In fact, in mouse studies, it was found that mice that don't have their bacterial flora, so to speak, right? So, their microbiome is non-existent. They do not develop certain types of T cells that are critical for protection against diseases. So essentially, bacteria that populate this microbiome that we carry with us, it trains our innate immune system, and the development of innate immunity by newborns, be it animals or humans, it is done based on the microbiome, and it's been shown time and again that the specific types of bacteria that populate our gut are almost genetically coded. Because if people are treated with antibiotics to wipe out their microbiome, a few months later they repopulate their gut with exactly the same bacteria. So it looks like there is a kind of synergy, kind of symbiosis between us and bacteria. And to some degree, the same is true for viruses, because there are a lot of viruses, for instance, enteroviruses that also populate our gut. There are countless numbers of these viruses, serologically different, and poliovirus, from which oral polio vaccine is made, the one that we proposed, it's also an enterovirus. And rhinoviruses that cause common cold are also enteroviruses. And the very interesting characteristics of these viruses is that they mostly produce asymptomatic infection. In fact, they were discovered in the 1950s when Albert Sabin was studying his new oral polio vaccine, and they found that there are viruses that they can isolate in cell culture, but they don't cause any disease. So they call them ecoviruses. It's called enteric cytopathic human orphans. Orphans because they don't have a disease. They also call them a virus in search of a disease, because, I mean, it just is completely asymptomatic. But when it infects you, it stimulates interferon, and it basically boosts up your non-specific innate immunity that actually protects you from other viruses, but it also helps you to clear newly forming cancer cells. And this periodic, maybe once or twice a year, infection with these viruses basically helps us maintain our health in a broad sense of the word. In a way, it could be not microbiome, but a human virome that is also is in symbiotic relationship with us. So I think that this is a very important concept, because in fact, it's not only enteroviruses. Coronaviruses, by the way, is the same thing, because we have 4 coronaviruses that cause common cold. And there was a study recently that demonstrated that we are infected with these viruses roughly 2 or 3 times every year. And the study was conducted in a very interesting way. I mean, they had a collection of sera from a set of patients that were studying for something completely unrelated. But for the study, they had to collect a sera from these patients for several years in a row. So they had sera taken every couple of months, from the same individual. So, and they determined the antibodies to these 4 coronaviruses, and as a control looked at the antibodies to measles virus. And the level of measles antibodies was flat. I mean, it was stayed absolutely level. But the antibodies to those 4 coronaviruses, they spiked. All of a sudden, without any disease, it would spike up and then go down. Then a few months later, spike up. And in the meantime, another one was doing it out of phase. So essentially, we are getting infected with these viruses without even knowing it all the time. And I don't think we can discount the effects that this asymptomatic infection produces in us. So I think it is a really very important, completely unappreciated concept.
Lots of really rich ideas here. Definitely, if other people want to jump in and ask questions, feel free to. I see a hand up in Holly's room, and we actually haven't met. Hello.
Hi, yeah, I'm Holly's husband, and I was a horse veterinarian in background, and got into virology and epidemiology through my involvement with the first recognition of Australian bat lyssavirus, which is our version of rabies in horses in 2013, and that's led into, ever since, I've been sort of researching Hendra virus and other zoonoses. And yeah, this is a really interesting group, and this is a great interest of mine, but one that I haven't had time to focus on while we're working on identifying the new strains of Hendra virus and things like that. But it's popping up in my research, for example, in the serology. So I'm looking at horses, I'm looking at humans and bats, and we're seeing massive cross-reactivity between, we've got a 33-plex panel, Chris Broder, who Konstantin probably knows in the US, is partnered with us, and we've got a lot of novel proteins on that serology panel, and we're seeing a lot of cross-reactivity. We're seeing sort of upregulation of reactions to other RNA virus families, particularly with our IgM assays. I mean, the IgM molecules is designed to do that. And then this is also with the human population, 700 humans that are veterinarians, a high-risk group, and then some comparisons in there. I'm seeing a much higher sort of likelihood of a veterinarian, regularly doing rectal palpations on animals with copious, pathogens everywhere, and having to stick horses down noses down horses' tubes and things, getting covered in. So they have a high exposure to a broad range of mammalian infectious agents. And I think it all adds up for me. I mean, it's also when you see the low levels of disease in rural Laos, for example, we don't know if they're not picking it up, but people that live with animals, that are interacting with nature, that are not in production, high-end production chains and very artificial environments, have a nice range of exposures, and it's helping. And I think two things pop out for me in my having met different virologists. Basically, I completely believe that we're seeing lots of evidence. For example, the repopulating, I think it was mentioned, of the microbiome of the gut as part of treatment for various problems. Reverse poo donor, therapeutic reverse poo implantation, which we do in horses, animals too now, not horses, small animals, but it's very common in humans now. The use of intestinal parasites, worms, to redirect immunity and improve inflammatory bowel conditions and this sort of thing. I work also with Jim Crow from Vanderbilt, where we're sort of making monoclonal antibodies for Hendra virus and Nipah virus. And Jim Crow's got a very interesting study looking at the immunome, so massive bioinformatical study, mapping all the immune immunoglobulin genes for a broad population. I think the problem is it's a Nashville population. It may not have as many sort of people from the tropics, but in time, when you start to get these sorts of studies coming up, we're going to know a lot more about the benefit from all these immune cells and the other from all these different pathways of immunity and the cross-reactive and adaptive immunity. And the other person is Gary Lynch, who Konstantin may have heard of. He's worked with Chris Broder and others in the past. And Gary Lynch is at Sydney these days, but he's got some interesting work on heterosubtypic immunity. So, in the context of flu, where you've got a lot of drift of the antigens of most interest, and they've sort of shown, well, there are some of these antibodies that are less important, but are highly conserved, and we're getting cross-reactivity. And he's also been working for years on this concept of taking broad immunity from the whole population as a therapeutic. So I think we've got I'd like to hear what Konstantin thinks of monoclonal antibodies and how they fit into his picture. And also, it'd be great to see him joined up with some of these other interests that I think are a bit similar, like Gary Lynch and the immunome.
Yeah, no, you're right. There are some people who explore this broadly cross-reactive antibodies. And just recently, last week, I saw a preprint of a paper that claimed that there is a cross-reactivity between RNA-dependent RNA polymerases of coronavirus and poliovirus, which they actually demonstrate by Western blots with a monoclonal antibody. And they actually make suggestion that this could be the potential way of using polio vaccine for preventing coronavirus disease. A totally different idea from ours, because we did not think about broadly protective antibodies, but rather about more innate immunity mechanisms.
Yeah, I guess what I'm saying, Konstantin, is I think it's important to, when I hear your argument, one possible criticism, which I think you should be ready for, and which you of course realize, is that there is, if we're going to just take these bits of evidence of people responding better when they have these particular genes switched on, we must not forget some of this other context, like the rapidity of the IgM response and the massive broad cross-reactivity of the IgM molecule, and also the cross-reactivity of other antibodies. I think and also with this sort of short-lasting, another part of the evidence is the short-lasting aspect of infections from common colds. And I guess from a cross-species perspective, as a veterinarian, it would seem in a horse, for example, the short-lasting and minimal infection response to a common pathogen or type of pathogen, viral pathogen in a horse, usually relates to it having seen it before when it was a young horse. And in that's that really special time when the immunity from the colostrum wears off around 3-4 months, but the immunity of the individual animal hasn't developed yet. And I think that would be a really interesting period to look hard with your work, because it's that innate immunity that's building in that time, building its strength. And but later in life, as an adult animal, when they're just pushing these things, having these infections come and go, I totally agree. I think there's a positive aspect to it, massively positive. 7% of our own genome is supposed to be viral. every liter of seawater is supposed to have 1,000 viruses in it. Every bit of lettuce we eat's got viruses from all sorts of animals in them. They are, in my opinion, one of the key missing links for Darwin that can switch on and switch off genes that interact with us. And we're getting evidence now that even the line between bacteria and viruses may be not as clearly distinct as we know, with sort of megaviruses, so that have been now discovered. And the age of metatranscriptomic next generation sequencing is showing us that we're still only dipping our hands in the water of a full knowledge of what's out there. So I think that's the only aspect that is important, is to not as always try to be very aware of some of these other holistic, broader contexts. And I think the big interesting thing for me that I really had no idea about was the some of this concept of the loosening of the chromatin in the cell, and the potential long-lasting benefit that's been mapped, for 9 months, for example, of an innate immunity. But being careful for some of those other bits of anecdotal evidence where people will come with criticism to say, oh, but there is, IgM, there is cross-reactive immunity to heterosubtypic, epitopes.
Yeah, no, I mean, you're absolutely right, but my position is that whatever works. I mean, there are multiple mechanisms that may be in play here. And my point is that we need to study this, because this, again, broadly cross-reactive adaptive immunity, such as antibodies, I think it may be real. It can be something that, just like everybody wants to have very specific and very antibodies with a very tight binding, right? But we know very little about those antibodies that are present in huge amounts, but are not strong binders. They can also provide some level of protection that is probably very hard to measure in vitro. I mean, and we know very little about this. So this can be something related to you were saying about this cross-reactive antibodies. Actually, I'm very intrigued by the question from Amina about the neutralizing the idea of infection. I mean, could you clarify what you mean by that?
Well, I love the way all of your proposals muddy the lines. So I'm we're speaking about vaccinations, which are anti-disease objects in the world, or artifacts, but we're speaking about them in terms of probiotics. And then you used the word infection, and it strikes me that so long as we think of a virus infecting, even if it's bringing us gifts, helping us be human, the word infection is so heavily laced with negative connotations that I wonder about that language. Is it more likely that we can reframe the language of infection and befriend it to be something that I got infected, or in the way we think about building immunity through infection, or do we think of probiotic infections more in terms of probiotic vaccines? So the language of vaccine and infection is an interesting dichotomy. Yeah, it leaves me curious. It also leaves me, as you're speaking, curious about just the language of disease itself. All of a sudden, the disease isn't, I'm not so interested in the seed as you're talking, the seed of disease, but more in the soil of the body as being the real hub or reality of an infection or of a disease, a phenomenon of disease. So I'm wondering if your own conceptions of, gosh, I just wonder if you would say, what do you think of diseases?
Well, look, I mean, I never thought about this way, about terminology, but you may be right. Maybe we should keep the word infection only for bad viruses, and the viruses that we think are good, maybe we should use the word vaccination. So I say, okay, I vaccinated myself with the rhinovirus. So, I mean, Yeah, no, there's, of course, I think that, most people think about viruses as something really, really bad. I mean, but we know that it's not true, I mean, because there are viruses, just like I described, that could provide a lot of benefits. And some of their ability to provide benefits is now being explored. For instance, you probably know about a lot of different ways to harness viruses for oncotherapy. I mean, there are oncolytic viruses, and there are companies that develop cancer therapies based on viruses. So this is one way of using viruses for the benefit of humankind.
Yeah, I mean, you're these pennies dropping for you is exciting to see, because it's something that I've kept coming up against. For example, I did some expert consultancy to a class action in the federal court against the Hendra vaccine, where it became very clear that what we call pharmacovigilance, which is what looks for the safety of and communicates the safety back to the community, it became very clear that there was a massive problem, that there was not sufficient vaccine bespoke pharmacovigilance in veterinary, where there's been 20 years of effort on this in the World Health Organization in humans. When we use a vaccine, you're giving it to an otherwise healthy animal. People think it's healthy. It might have 5 active infections at that time, but they're subclinical. It might have a lot of other causes of disease. Horses love to kill themselves, put their foot through a fence or something, or react in dramatically in the environments that they're often in. They're often getting
actually I might intervene here, and we've only got about 5 minutes, and I want to give Halia a chance to ask her question as well. And I wonder, so Halia, you've got a question about lifestyle in there, and I wonder if it's related to some of the things that I've been thinking. There's long histories of ways of speaking and thinking about nature, about being alienated from nature, and kind of the modern condition is one where we're not interacting with the sorts of wildlife that we were just hearing about through the veterinary practice. Were you going to ask something along those lines?
Yeah, I was just going to ask if you had any thoughts on, I guess, in anthropology, regularly come across medical researchers who are interested in the question of whether the disease profile we have in the West is due to a particular kind of lifestyle, specifically one that's obsessed with cleanliness and raising children in sterile environments and that sort of thing. It's, of course, it's just there's so many factors, so it's very difficult to isolate out anyone as the cause. There's nutrition, there's a mother's literacy, there's so many factors that can affect the health of a child growing up. It's hard to pinpoint, this or that. But I was wondering if you had any thoughts on
absolutely.
lifestyle.
No, I think you're really right on target with this question, because I was also thinking along the same lines, because when this pandemic started, especially in last year, a lot of people were asking, how come there is such a remarkable correlation between the COVID incidence and the gross domestic product and the human development index? It's just fantastic, So it's some people even call it how do they call it? Clean hands disease. Basically, that countries that don't have a perfect hygiene and in general public health, and so they tend to have less
that's right
virus. And in some reports from countries in Africa suggest that a significant part of the population are already immune to this virus. So basically, this pandemic went through some of the countries without them even realizing that they have a problem. So of course there was some incidents, but by far lower, with much less severe disease and mortality. I don't know if it's time to actually do the final analysis. It still holds true, even though in the last few weeks we're seeing that those countries that held pretty well against, like for instance, Southeast Asia sees the disease on the rise, and nobody knows why. Why did they wait so long? But still, even with this increase that we are seeing, it is orders of magnitude lower than we had it in the United States and Europe. So there is some reason. Some people suggested that there may be circulation of a related virus, that populations in these countries are partially immune because of the cross-reactivity of with other viruses. But it well may be that, you know, again, my explanation, and I'm maybe a little bit biased here. I thought that perhaps it's based on the circulation of other pathogens that constantly train and keep innate immunity in these countries up on high alert. So that's why they, and there are some data suggesting that this may be the case, because, for instance, in these countries, the efficacy of live vaccines given orally, such as polio vaccine or rotavirus vaccine, is very, very low. And the explanation is that there is a strong interference. There's so much, you know, gut infestation with other pathogens and bacteria that basically the vaccine doesn't take. It just completely is not effective. So this could be one of the, and this kind of clean lifestyle that we have, it has negative impact on some other things as well, as you know, like asthma is almost non-existent, and other allergies are non-existent in countries with the suboptimal hygiene. So I mean, it's a kind of trade-off between clean lifestyle and health.
Yeah, this is super interesting, and you know, I think also speaks to some longstanding concerns in Rob Dunn's lab, where Amina is based. You know, Rob has a book that chronicles all sorts of different things that we've lost as part of the modern condition, all sorts of what we might call commensal or symbiotic, or in the language of multi-species studies, we talk about companion species, creatures that kind of make us who we are. And vice versa. So I think, you know, these logics of Pasteur and others who, you know, isolated microbes, gave us a new way of visualizing and thinking about things like contagion and infection. You know, they do work in particular situated contexts. You know, if you want to avoid dying from bacteria when you're an infant, you know, it's pretty good idea not to expose yourself to cholera, or, you know, I'm sure there's measles is another one. There's a village that I visited in the highlands of New Guinea in West Papua a few years ago, where a measles outbreak recently killed, you know, 56 children. But at the same time, these pathogens are there. I think it's the riotous diversity of this undescribed universe of the human virome that your work is starting to illuminate as part of what keeps us healthy and you know, functioning in these ecological conditions. So we've got just a minute or two left. I was wondering, do you have any final thoughts before we wrap up today?
No, I think, I mean, my main point is that this is clearly a field that deserves much more attention that currently is being paid to. And I was a little, I was happy to learn that, for instance, even though NIH did not fund our study, at the same time they decided to have a workshop that will take place next month to discuss exactly these points. So, you know, this huge ship of government-funded science, it turns very slowly. And I think that I see that my goal in this whole episode is just basically to raise awareness that this field exists. It has a huge potential to improve human health and to prepare us for the next pandemic, and perhaps it can generate a lot of other interesting therapies for maybe not even infectious diseases, but things such as cancer and other conditions. And I also want to thank you for inviting me. It was really interesting format. I've never participated.
Well, thank you so much, Konstantin, and I hope this is the first of many conversations. And yeah, someday in a post-pandemic world when travel is possible, I'd love to meet. So we'll circulate your new article that you've just shared with us, and that workshop also sounds super interesting. So thanks again for joining us, and hope to see you either in a Zoom room or somewhere else soon.
Okay, all right, guys.
Thanks so much.
All right, bye-bye.